Good morning. Today we are taking our notes over the phases of mitosis. You need to make sure you have your levels of organization here. I've highlighted cell because that's what we're working with today. You need to make sure you have your Cornell notes down here on the side. This is 10 points, this is 5 points. Alright, so we're going to start with this box here where we remember the phases by the acronym PMAT. Okay, so that stands for Prophase, Metaphase, Anaphase, Telophase. Another way you can remember is it's prime, middle, apart, and two. So PMAT, Prophase, Metaphase, Anaphase, Telophase, prime, middle, apart, and two. Okay? So mitosis is when a cell separates its two sets. It has two sets of DNA, two complete sets. In preparation, this is preparation, to make two new daughter cells, okay? It's important that you remember that it's two sets of DNA because we're going to divide them in half. Half is going to go this way, half is going to go this way, and it's going to form two daughter cells that each have their own complete single set of instructions, which is DNA. Okay, so DNA are instructions. Each cell has to have its own. So before they split, you have to have two sets. So this one gets one and this one gets one. Otherwise, you would just have half and half, and then you would die because you only have half of your instructions. All right. So we're going to go through the phases here. The first phase is prophase. It's the first phase and it's the longest phase. This is when chromatin bundle up. So chromatin are the long strands of DNA. They're going to bundle up forming chromosomes, okay? So these are really long and stringy like spaghetti, and it's hard to divide those because they're just all over the place. They wind around a protein to become very compact and small, so that way it's easier to separate them later, okay? So chromatin are long and stringy. Chromosomes are wound up real tight like a spring. All right, sister chromatids, which chromatid is the plural of chromatin, they're joined at a centromere, which is basically in the middle. So what this looks like is have your chromatin here. Okay, so let's say you have your sister chromatin. They've been wound up into very manageable sized chromosomes, right? They're all twisted real small. It'd be a lot easier if you were a cell. Okay, so they're twisted up. We've made our chromosomes. Now these two are the same. These don't look the same, but we're pretending they're the same. They're going to be joined together at a point in the middle to keep them paired up. So you know that every cell is going to get the exact correct number of instructions. So these two are both red, so they're going to pair up together. So let's say we had some blue ones, then the blue ones would pair together, and then if we had green ones, those green ones would pair together. Okay, so they're keeping like together and they're stuck together in the middle by something called a centromere. Okay, that's why we get this little familiar X shape. All right, so there are two other things called centrioles, and they move to opposite sides of the cell. They are where the fibers are going to come from to pull the chromosomes apart. Okay? So metaphase, M is for middle. It is very, very short. This is where the chromosomes line up down the middle of the cell, right down the middle. Right? Then anaphase. The sister chromatids are pulled apart. A is for apart, M is for middle, A is apart. So we have our chromosomes. It's very confusing, I know. So we have our chromosomes. They are lined up down the middle of the cell, right? Now during anaphase, they're going to be pulled apart and go to opposite sides of the cell. These are now called daughter chromatids. They were sisters, Now they're daughters. Okay, sisters have daughters. Spindle fibers pull those daughter chromatids to each side of the cell. So the cell needs, one cell needs this one and one cell needs this one, so they're going to get pulled apart so the cell can then divide right down the middle. All right, telophase is the very last phase. You're going to make 2 new nuclei, so T is for 2. 2 new nuclei are formed, and this is when cytokinesis begins. Okay, so telophase is the end of mitosis, the beginning of cytokinesis. So you have a cell, and it starts to cleave like this. This up here is called a cleavage furrow, and down here, cleavage furrow. It starts to cleave, and goes like this, and then pops apart. And now you've got 2 brand new cells. Each has its own set of DNA. Each can function on its own. It doesn't need help from anyone else. And it's going to go back into interphase. And it's going to grow and grow and grow until later, when these 2 need to divide, they can. Okay? So the only thing mitosis is doing, this whole process, is how chromosomes bundle up. They stick to each other, they get pulled apart. It's like how cells organize DNA to move, basically. If you and your boyfriend break up and you're gonna take your stuff and he's gonna take his stuff, you're gonna get it together and you're gonna decide what goes where, okay? And each of you are gonna take half. So in your notes down here, chromatid is the plural of chromatin. These are very confusing words. I know, that's why we're writing them down over here. Sisters make daughters. So when we talk about sister chromatids, when they divide, they're now called daughters. The 2 cells, after the cell divides, those are daughter cells. Sisters make daughters. And centrioles are what send out spindle fibers, okay? So when you get done with these notes, we're going to have a slightly in-depth foldable that I'm going to have another video to show you how to do. Thanks for watching.